Okay, so by far the most common things I saw that were requested to review were histology slash skin system, the skull, and there were a couple other areas I can touch on with the skeletal system that tend to trip people up. And then really the only other thing is just general orientation. Like when you get to a lab exam question, you'll be switching stations every 75 seconds. So what are some tips to not freak out and like actually figure out the view you're seeing stuff from? So if there's anything you want to add, we can absolutely go over that. Or otherwise I can start with histology.
That is fine.
Okay. So as mentioned in class, I will write my lab exam questions based on my PowerPoints. The PowerPoints, they take information from a lab manual, so it's all connected, but the way I would recommend reviewing is to actually use the PowerPoints. So there's those gold bolded terms starting with the skeletal unit that I would just make a running list of so that you can make kind of your own answer key. And we also can't forget about our cytology, histology, and the integument labs. So the stuff in there is still important, but it is not highlighted. And that is because this stuff is really need to know information. As you've probably figured out from the next several labs, as we talk about connective tissue and epithelium pretty much with every lab. Some exceptions are like when we just go over appendicular skeleton, but all this stuff is need to know, which is why I have not highlighted stuff in here. But all those hints I've given in class of like, this would make a really good lab exam question, or this one is pretty easy to find under a microscope. That is a good indication that a certain tissue is more likely to appear on the lab exam. So I'm really trying to give as many hints as I can, but I'll go over the things that I think are most important to know about histology. In addition to the PowerPoints, I highly recommend using those histology resources under the resources module. So the one that we use the most right now is Yale School of Medicine. If I want to find a nice photo of histology, I usually check out that Yale site first because they have nicely labeled images, unlabeled stuff, and that's another really good way to study. So if you're struggling with connective tissue, which many people are, you can go to the corresponding lab and then scroll down to our slides here. And, I don't have a great example of dense irregular connective tissue on Canvas. So I might come in here. This is what it looks like. I might take a snip of this and use that. So these kind of slides that we have on the Yale site are the gold standard for histology questions. And that applies to the microscope stuff too. So I will try to find example slides when I'm setting up the microscopes that look like these Yale examples, because I think these are some of the best examples that you can find online for our lab. So with that said, I'm just going to draw some information from the PowerPoint here and we'll switch back and forth to that Yale site. So let's start with epithelium versus histology, or sorry, epithelium versus connective tissue. So just probably the main thing to know here is that our epithelium is mostly cellular. We're mostly dealing with cells and just a little bit of matrix, whereas our connective tissue is mostly matrix. We'll see a lot of collagen fibers and other proteins, but you're not going to see a lot of cells. That is one of your first clues you can use when you're trying to identify epithelium versus connective tissue under a microscope. And we'll see that applies to every single type of epithelium we've talked about, stratified versus simple, all that good stuff. If you are not sure if you're looking at epithelium, look for nuclei. So in these examples here, we're zoomed in. This is an example of columnar epithelium, but each of these guys here has that dark purple nucleus. Same thing over here. This looks like pseudostratified ciliated columnar epithelium. We still have those layers and nuclei. So that's a really important thing to look for. If you're not sure, if you're second guessing yourself, if you're dealing with epithelium, look for those nuclei. Connective tissue is a little messier. So I'm gonna gloss over skeletal and nervous tissue right now because I think most people are pretty solid on those. This stuff is where it gets really confusing. So with connective tissue, the two things we want to look at are pattern. Is there a noticeable pattern? Are we dealing with some sort of wavy, regular pattern? Or are we dealing with something like this, where we've got these fibers running every which way, creating this kind of cobwebby appearance? Depending on that, you can tell whether you're dealing with regular versus irregular connective tissue. We can also look at the density. So our common dense types of connective tissue are either dense regular or dense irregular. But in both of these cases, there's not really any white space between the fibers. It's densely packed together. So we're either dealing like with a rope-like pattern or just this really thick web of connective tissue. So those are the main clues I use to tell apart dense connective tissue from our other types. So I talked a little bit about reticular connective tissue, but I did mention in class, this guy is associated with organs. So we don't really see it isolated on its own. We don't have a great microscope question or a great microscope slide example in class, so it's hard to ask that one on the exam. But things like our loose areolar connective tissue are very distinct. They do make a nice lab exam question, whether that's PowerPoint versus a microscope question. So anything like this where I've pointed out, this is a really distinct type of adipose tissue. It's easy to tell apart from the other stuff. That's really a good hint that this might show up on the lab exam versus something like our reticular connective tissue. So those kind of hints directly apply to the lab exam too. We did talk a lot about the different types of cartilage. So make sure you know hyaline versus fibrocartilage versus elastic. And this is where we can use those context clues from the PowerPoint to help. So when I'm writing my questions, I will literally take phrases from the PowerPoint. I might say, identify this type of cartilage which reduces friction at joints. We see that exact term right here. So if you are familiar with the PowerPoints, if you really know your hyaline cartilage and know where it's found, that can help you. You can almost answer the question without having been to that microscope slide yet. So that's how I would use these PowerPoints. All these examples in here, these visual examples, are what to expect under a microscope. I'm not going to pull any surprises on you, but you can also use this extra information as context clues to help you answer the questions. Same thing with spongy versus compact. So we spent a lot of time in that attendance sheet figuring out what you can see on our bone model from that spongy versus compact view, as well as what you can see under a microscope. So we talked a ton about this unit called the osteon within compact bone and everything that's going on within that like tree ring pattern. So for example, since I mentioned that the osteon looks like a tree ring pattern or a tree trunk, it might not be as much of a surprise on the lab exam if you see that tree ring phrase come up. So those hints I use in class are things I will use to word the questions. Yeah, so those are the main things. Epithelium does mix people up. So I would spend a lot of time on epithelium, just on the difference between simple versus stratified stuff, specifically under a microscope. So these types of diagrams are useful when you're first learning the information, but I really think people will benefit from using the Yale site when they're studying their epithelium. So I'll go back and give you an example of that. So we're in epithelium, scroll down to our slides here. So this is a really nice example of our simple cuboidal epithelium. But if you don't know what to look for, this whole slide is bewildering. We've got this stuff over here, which might be connective tissue. We've got like other passageways here. So it's like, what do you even focus on? Well, on the microscope questions, there is that pointer on the microscope. So whatever I'm trying to ask you to identify, I will place that pointer on one of those cells or structures. So we would have a little more context here. Maybe I want to point to this cell here and say, identify this type of epithelium. And maybe I'll give an example of where it's found, like in the kidney tubules or something. So you'll have the pointers to help you, but you need to be comfortable taking a slide like this that is not nicely labeled and focusing your attention to just what is under the pointer. So don't let this other information throw you off. Here's another example. So a lot of times when we're dealing with columnar epithelium, it's in some sort of digestive tract lining. So when we take a slide like this, we might zoom out a little bit, and I can actually show you that from a real GI tract. So a lot of times our slides will look kind of like this from a 4x objective lens. So when I zoom in to try to figure out a question to ask you about that columnar epithelium, it might look a little more like this and less like this nicely zoomed in slide here. It's just depending on what I can find out of the microscope. So you need to be comfortable kind of filtering through this information, just looking at where the pointer is going to be to help identify this type of epithelium. So for that, I think it just benefits to practice with a lot of different slides to just practice that orientation. All right, any questions on histology, epithelium, connective tissue?
Do we need to know, I don't know if you can tell me this, but do we need to know the difference between the elastin fibers and the cartilage fibers?
Good question. So I will say that elastic fiber is a pretty good question to ask because it is really distinct. Anytime we have elastin, it shows up really dark on slides. Let me find that in here. And you got to be careful too, because we have two types. We've got this elastic connective tissue, but we also have elastic cartilage. So I think it'd be really helpful if you know the three types of cartilage, because this is a really nice example where we've got these dark elastic fibers. It is a little trickier to see with elastic connective tissue. Let me go back up here. Are you going to take me back up? Okay. So that was cartilage. When we have elastic connective tissue, dense connective tissue, this is usually found in combination with other types of connective tissue. So one really good example is along the lining of blood vessels. That said, it's a little messier under a microscope, so hopefully that gives you an idea of which type you should focus on, the dense connective versus the cartilage. Okay. If we have time, I'll go over the skin histology at the end. I think that one's a little more straightforward. There's that recording as well that you can rewatch of the lecture for integument, which I think covers the main points to focus on for the exam. Next, we can talk about orientation with the skeleton. So let me just search. I'm going to search for some. I'm debating searching for bones, or we can also look at our scaly images. Let's try that first. So at the bottom of the skeletal system unit, we have these articulated skeleton images. So they're called skelly photos, articulated skeletal system. This is useful to practice orientation. So I think not like 90% of the time, I will give you questions in anatomical position, meaning like the arm is supinated, you're facing forward, looking at an anterior view of the body. There are a couple of circumstances where I need to get at some of those posterior landmarks on bones. So it's really important to always figure out if you're looking at an anterior versus a posterior view of whatever skeletal unit you're talking about. So these are going to help. For example, we have different views of the elbow. That is one that tends to trip people up. Oh, these are very big. See if we can get that a little smaller. Here we are. So it's really important to know whether you're looking at an anterior view or a posterior view. Now, any question I ask on the exam about something like the humerus or the radius or the ulna, you're going to have the other bones of the elbow joint there to help orient yourself. I'm never just going to give you a radius and ask you to identify stuff. It'll always have the other bones with it to help you with this process of orientation. So in this case, we have an anterior view. And the way I can tell is I look for that olecranon fossa. That is going to be a really huge fossa that is posterior. So if I don't see that guy, or if I don't see this olecranon process fitting into that fossa, that tells me we have to be in an anterior view. So I'll try to put these side by side so we can compare them. Maybe. Close enough. Okay. So if you have this kind of view of the elbow, where you've got that really large process coming off the ulna, that's going to be your olecranon process fitting into the olecranon fossa here. That's how you can tell you're in a posterior view. If we don't see that, if instead we see these nice views of our condyles, in this case, we've got that kind of hourglass shaped trochlea and then the rounder capitulum above the radius, those are good clues that you're in an anterior position. So when you get a question like this, always start with orientation. Like no matter what the question is asking, forget about the landmarks before you figure out if this is an anterior versus a posterior view. Once you're there, it might take like 30 seconds if you have a weird view, but take that time. So once you've got that, you can start to identify landmarks. This is where it's really helpful to study the bones from different views. So specifically look for an anterior view of the elbow, figure out what landmarks you can see from that view, and then do the same thing for the posterior view. And you can kind of keep separate lists of what you see in each view. That's going to help you keep them separate on the lab exam. But those are two ways I'd orient to the elbow joint, or if you're given a humerus question, radius, ulna, this is all going to be proximal, radius, and ulna. And this may be helpful for your studying as well. I tend to focus on bony landmarks that are found at joints, so at the ends of bones. So we talked a lot about the elbow joint, we talked about the knee joint, the hip joint. So I would focus your studying on the kinds of landmarks you can see there at these busier areas, rather than like along the shafts of the bones. The femur is another really good one to know because we have just so many landmarks that I could ask you at both the distal and the proximal end. So for example, we can take a look at our knee. I'll just go ahead and open up two different views for us. Okay. So that's our anterior view, and we need posterior view. Okay. So here we have them side by side again. What I do is take that list from the PowerPoint and just check off what you can see from this posterior view versus that. In this case, we're looking at it from an angle, like an anterolateral view, but we can see those most important structures on the anterior side. So same thing here. You may not have this patella, but you will have a femur, a tibia, and a fibula in anatomical position. If I want to get at the posterior bone, I will, of course, have to flip everything over, but it still will be oriented so that you can figure out your left versus your right side. So what I mean by that is your fibula is always going to be lateral. Your tibia is always going to be medial. So in this case, it's going to be beneficial to figure out lateral versus medial. And then you can figure out if you're looking at the back of the knee or the front of the knee. The best way to tell is look at your femur here. This is our posterior view, and this is actually the best view from which to see the condyles of the femur. In this case, we have our lateral condyle above the fibula, and we have a medial condyle above the tibia. Those smooth parts are the condyles, and those are always best seen posteriorly. Versus this view, we don't really see those. This is a better view for things like our epicondyles, those little ridges above the condyles. Another clue you can use to tell your anterior is if you see this bump, this is that tibial tuberosity, that connecting point for the patellar tendon. So it comes over the patella, or patella is kind of embedded in it, attaches down here. So those are two clues you can use to tell apart anterior from posterior. Again, do that before you try to figure out your landmarks, because otherwise you may be wasting your time if you don't know you're in the right view. This is also relevant for those knee questions. So last lab, we talked about the knee joint. We have some separate images for that guy. So under lab 9, I would also use all of these views here. So I'm not trying to be too mean to you guys. I'll mostly ask stuff from pretty straightforward anterior, posterior views. Go ahead, take a look at these. So in this case, there's a lot of soft tissue questions I can ask too, but the same kind of orientation tips apply. Get out of these views. Okay. Oops. Did not mean to do that. Okay, so here we have it again. Exact same rules for lateral versus medial apply here. So once you get a knee model like this, figure out your lateral versus your medial. Again, the fibula, the smaller bone here, is always lateral. Tibia is always medial. From there, you figure out if you're in a posterior versus anterior view. Again, we have these condyles to help us. The femoral condyles are always best seen from that posterior view. In this case, with the joint model, we also have the patella to help us. So that's a big clue. This is going to be that anterior view. This is important because all of these soft tissue structures we talked about, all the tendons and the ligaments and the menisci, those are named directionally. So you absolutely need to know tibial side from fibula. Same thing here, because, for example, your lateral collateral ligament runs with the fibula. Sometimes you'll see it called the fibular collateral ligament. So these things are easy to mix up if you don't take that time to orient yourself. Another thing that is really confusing for students is when we get to the cruciate ligaments. So those are the guys that cross underneath and kind of through the knee joint, the deepest one here. If you are in a posterior view, one helpful tip is that our posterior cruciate ligament, that PCL, is first of all more prominent from this view, but it also usually has fibers running into our lateral meniscus, which is this kind of wedge here. That's a really good clue that this is your PCL. So that number 9 right here would be PCL. Anteriorly, the ligament that predominates is going to be that ACL, the anterior cruciate ligament. So if you know you're in anterior view and you get a question about this guy here, just say ACL and that would get you full credit. You can abbreviate. So hopefully those give you some ideas of how to orient. The one other problem area for students is the pelvis.
I have a question.
Yeah.
The joints are going to be on this test.
Just
Or
the, sorry, the knee joint is going to be on the test.
Okay. But the
So,
rest of the ones from like our most recent lab won't be.
so we talked a little bit about the elbow, a little bit about the hip, shoulder, but the stuff in gold only pertains to the knee joint. So that's the only thing you can expect to see.
Gotcha. Thank you.
All right, last thing I want to look at for skeletal stuff is, before we get to the skull, that is, it's going to be our pelvis. Okay, there's one view, and then we also need this view. Actually, we're going to look up if I can type here, we're going to look up a pelvis model. Pelvis is tricky because sometimes I can ask you from an articulated view like this, but I can also ask you if we just have a single os coxa disarticulated. Let's try this. No, I need to specify my view as well. Lateral view. Okay, here we go. Let's start with this articulated view. So we do have a couple models out there in the lab room. I'd recommend taking a look at them Tuesday with this articulated pelvis. So we got one os coxa, two os coxa. And what tends to trip people up is all of these spines along the ilium. So the first thing you should do always with bone questions is orient yourself. It's pretty easy to tell that we're in that anterior view because we can see our pubis here. We can see that pubic symphysis. And generally, you get this nice bowl shape when the pelvis is put together. I would start up top. Most of the landmarks we talked about that were highlighted in the lecture were associated with this ilium. So this top half of the pelvis, kind of above the level of the acetabulum, this is going to be all ilium. I would start with the iliac crest. That's this most prominent ridge that you see here, because we have two landmarks at the terminal ends, posteriorly and anteriorly. So in this view, this point where the iliac crest ends, that's going to be your anterior superior iliac spine. Remember, the first two terms are directional, anterior and superior, and then each of these pairs, these four terms, they're going to end with iliac spine because they're like little points. So we have that ASIS. We're going to have the AIIS a little lower. This one is tricky to see from this view, but you can see it outlined. The red here is where we have muscle attachments. So we're going to have a specific muscle attaching to that AIIS. And that's all we can see in terms of spines. The other two will be posterior. We also have a nice view of that iliac fossa. So don't forget about those shallow depressions. We talk a lot about these more prominent projections, but I can also ask about iliac fossa. Same goes for the scapula. We had 3 fossa associated with the scapula. So make sure you're paying attention to those in your studying. There's not too much I would ask about the pubic region. So we do have some bumps and lumps here, but I'm more focused on these important things, which are going to be attachment points for muscles later on. The one region in the ischium that I would pay attention to is best seen from that lateral view. So this is where it gets tricky for people. First, you need to know whether you're looking at a lateral versus medial view of the pelvis. So you might just have this guy laying on the table and not much else. So we need to use clues here, like the presence of this acetabulum, that cup-like depression. This is always going to be lateral because this is where the head of the femur fits into that hip joint. So it doesn't make sense for it to be medial. That just wouldn't work anatomically. So the acetabulum is going to be lateral. That tells us this is a lateral view of the pelvis. Once you get that, you need to figure out anterior versus posterior. So a couple clues we can use. The pubic region comes to a point. So I think P for point, P for pubic. So that pointier part is going to be more anterior. The ischium can also clue us in. So the ischium has this really large roughened area. That's going to be your ischial tuberosity. And that is like our seat bone. So seat bone is going to be posterior. That's like the most posterior part of our butt that we sit on. So those are 2 clues to tell anterior from our posterior ischial tuberosity. Last thing that you might notice is the ilium kind of arcs more dramatically towards the back. So if these don't help you, you're still unsure, follow that iliac crest back, and then we have this really dramatic kind of angle here posteriorly. So those are 3 clues you can use to help you. And then a lot of questions will pertain to those spines, those iliac spines. So we had ASIS. We're going to follow that back. And then this more rounded, but still kind of pointy projection, that's going to be your PSIS, posterior superior iliac spine. Immediately below that, we have another little lump that is going to be your posterior inferior iliac spine. If you ever get mixed up, which is common when you're under a time crunch, I would always follow the iliac crest to that superior landmark. And then the one immediately below it is the inferior counterpart. So I've used that to help me when I was kind of panicking on the lab exam. Follow this back. This has to be that superior landmark. This has to be PIIS. Whoa, PIIS. Don't know if that's any easier to say. A lot of times students do confuse this guy with this spine. So the only other really major landmark within the ischium, in addition to that ischial tuberosity, is this little spine here called the ischial spine. So don't confuse that with your PIIS. This is below the level of the acetabulum. So pretty much this is our dividing line between ischium and pubic region and that more superior ilium. So those are some clues to help you. Okay. And if, this is a lot of structures to talk about. So one good tip to keep in mind is if I am going to ask you a bony landmark question, I need to be able to put an arrow on the landmark and have you pretty clearly know what I'm asking about. So assuming you've studied, you know your landmarks really well, I should be able to put an arrow on something and people should know what that is referring to. So that means things like this guy here, this greater sciatic notch, those are a lot harder to ask about because I can't just put an arrow in this region and say, what is this? Now, something like the acetabulum is a little clearer. I can put an arrow in here and you should know that that I'm asking about that depression, that acetabulum. If we get an opening question, I usually run a wire through that opening. So I can put a wire on the arrow and say, identify this opening separating the pubis from the ischium, something like that. But these areas are trickier. So you can hopefully use that to help narrow down your list of structures to focus on. Any questions on appendicular skeleton?
I do not think so.
Okay. Sounds good to me. So the other major area we want to focus on today is the skull. So speaking of openings, this is where I will utilize wires to ask you about openings of the skull. Now we do have some nice images here, and I have mentioned this a little bit. I don't know if I made it explicitly clear, but I have a whole playlist that I use for my hybrid class. Some of the virtual labs include the appendicular skeleton. So these are totally optional viewing, but if you want more review with the models that we will specifically see on the lab exam, all of these links will take you to different regions of the appendicular skeleton. And then our skull lab was also virtual. So if we come down here, I think that was lab 6. Yeah, there's also YouTube videos for the skull going over all of these regions here. So that's another way to study. But the most common way that skull questions will be asked will be from an anterior view and then from an inferior view. I'll go ahead and pull up some of these and then we'll just search up an inferior view of the skull. Nice part is there's a wealth of resources if you just go to Google Images and search up a specific view of a specific region. So we're going to look up skull inferior view, unlabeled. And we want a real one for today. See if this guy works for us. Yep, this will do. Okay. So let's start with that interior view. So we spent a ton of time going over openings of the skull because there are a lot of really good candidates here. So I usually will pass a wire through openings if I want to ask you about them. So if you see a wire question, be prepared to answer some sort of foramina, canal, meatus. So again, you want to orient yourself before you try to answer anything. I think it's helpful to think of these in terms of our 3 cranial fossa. So kind of the first third of the skull here is that anterior fossa, and the dividing line here is that lesser wing of the sphenoid. I think it kind of looks like a manta ray. Some people say bat, butterfly, whatever makes sense to you. But this is going to be that line between the anterior and then our middle cranial fossa. And then basically this entire span of bone, kind of another hourglass shape, that's sphenoid bone. So this is the lesser wing, and then the greater wing is this part that's spanning the entire skull. This is where it gets really busy. So we talked a lot about our ROS foramen and L. So we can't see all of those super well here. I can point out O, L, and S. So kind of tucked underneath this lip here of the lesser wing, we would have our foramen rotundum, which would be like right here. Unfortunately, I can't show you that on this image, but that's where we start. We start ROS, and we're kind of making a lateral path here. On this side, it's a little clearer, so R would be just under that lip. Foramen ovale is that largest opening, which is oval shaped. So this guy here, really good lab exam question because it's easy to see when you're looking down on this view. Easy to put an arrow through. And then this would be our S. So unfortunately, on the models of the skull that aren't real, sometimes these foramen spinosum are not punched out completely, so it is hard to ask about that one. It's hard to put an arrow through here. So think about, common sense wise, can I pass an arrow or can I pass a wire through it or put an arrow on it? And that should help narrow down your studying. So that was ROS, and then we have L. Again, this guy looks a little different depending on if we have a model or a real skull. So either way, foramen lacerum will always border this central structure called the sella turcica. Kind of looks like a saddle. So if this is like the front of the saddle, this is the back, this is that dip in the middle. So bordering that or sandwiching it, we always have foramen lacerum. If we are dealing with a real skull, this foramen lacerum will look more jagged. It looks lacerated like a knife cut. So that's how I remember it. Other than that, be mindful that we also have this optic canal. So I would have to tilt this skull slightly, and we would go in here through that optic canal and come out in the orbit of the eye. And then possibly the only other thing I could ask you from up here is the superior orbital fissure. So again, we would have to come in through here. Can't see it from this view as well. But we have that superior orbital fissure on the back wall of the orbit. I do want to point out this stuff as well. So within the ethmoid bone, we have a couple of areas here. This vertical projection is that rooster's crest, that crista galli, and that is sitting on this plate here called the cribriform plate. There are little holes dotted in that plate called olfactory foramina, or sometimes you'll see cribriform foramina. Those are, we'll talk about it later, but they allow little fibers from the olfactory nerve to pass through. That's how the sense of smell gets to our brain. So that is everything in that anterior and middle cranial fossa. And that leaves this region here. So, there are some theoretical bounds here, like this rocky ledge looking thing is going to be that temporous portion. Sorry, I got that backwards. Petrous portion of our temporal bone. So petrous means rock-like. This is like a really hard part of the skull here. And then embedded in that, we have two openings that I want you to know. So we have the jugular foramen and then the internal acoustic meatus, or sometimes you'll see internal auditory meatus. Either term works. There's a lot of different names for it. So that guy is within that petrous portion, just under this lip here. A little further down, we have that jugular foramen. People mix those up all the time. So one way to tell them apart is the auditory meatus, that IAM, is higher up. It's more superior than our jugular foramen. This really dark opening you see would be that jugular foramen. So that's always closer to our foramen magnum, that biggest opening in the skull. So make sure you know those two and can tell them apart. The other opening, again, can't really see it here, but I can show you from a different perspective. We have this foramen magnum, and then just on the lip of that foramen magnum, we have our hypoglossal canal. So if you're on this level at the very bottom of the skull and I put a wire through here, it's asking about that hypoglossal canal. And I will try to find a different view to show you. Let's see if we get any. Okay, I think this will do. So this one is a little interesting. We are looking at the top here at the middle cranial fossa with that lesser wing of the sphenoid. And then we're looking all the way down at the foramen magnum here. So this shows those levels. We're kind of starting up on the first level. This is the second level. This is the basement, that posterior cranial fossa. So ignoring the ROS that we just talked about, the next structure down from the ROSL foramen is going to be that internal acoustic meatus, which would be like right here. It's not pointed out on this view, but what we do have is that jugular foramen. So you can see this is just superior to our foramen magnum. And then on the lip of that foramen magnum, we have the hypoglossal canal in green here. So that's a hot spot for lab exam questions between the middle cranial fossa and then those things near the foramen magnum. Last view is the inferior view. So I'm not going to go over, I mean, we can if you want to, but I was not planning to go over things like our sutures. So I'll just mention this, we can ask questions from this exterior view of things like the sutures, the bones of the cranium, some of the bones of the face. Again, in the interest of fairness, I will only ask questions about things that are easy to tell apart. So kind of the facial bones get a little questionable because they're kind of blended together, especially on our models. So study these, but I would pay close attention to your sutures and your bones of the cranial vault. And then maybe, there's a couple things I could ask you about maxilla or mandible. We did spend a lot of time going over landmarks of the mandible, but that hopefully gives you some direction for studying the exterior view. But the other tricky view for people is this view, the inferior view. So if I happen to put a skull in this view, it's because there are things in this view that you can't see easily from a different view. I will not ask you things like ROS from this view because we have a much easier way to look at those. But things that are unique to the inferior view would be these occipital condyles. This is the only way to view them. We also have some of these landmarks that are associated with the temporal bone. So we talked a lot about that TMJ joint, the temporomandibular joint. Well, we have this condyle here, that mandibular condyle, mastoid process. There we go, mixing it up. Mastoid process, that's that part just behind the ear that you can palpate on yourself. So that's a good one to ask. We also have styloid processes. So it's a little hard to see here, but this pointy part just in front of that process is going to be the styloid process. We have mandibular fossa as well, just posterior to that styloid process. And the other main thing here is we have this really nice view of the carotid canal. So the carotid canal takes a kind of tortuous path through the skull such that we can't see it from that interior view when we're looking down on the cranial fossa. And so it does not puncture directly through like all those other openings we talked about. So this is really the best view to see that carotid canal from. So between the condyles and the canal, and then those landmarks of the temporal bone, that hopefully gives you some hints. Are there any other areas of the skull that you are struggling with that you'd like to review?
No, I think I'm good.
Okay. So those are the main talking points I wanted to go over. If there's anything else, feel free to shout it out. Otherwise, I'll be on here until 4, just hanging out in case we have other people dropping in.
Thank you so much.
You are welcome, and good luck studying.